Hi, thanks everyone. Thank you so much for having me today. I am super honored to be here and present on behalf of AbbVie. A number of people outside of AbbVie who we've consulted with on the development of our spinal cord injury program, including Plex, which we did at the beginning, but not to the extent that we should have. I'm going to be up here saying we could have done things better, but now we're even more involved in the community. I want to thank everybody, and particularly those people who have enrolled and participated in our clinical trials, which I'll tell you about a little bit today. So first of all, a little bit about AbbVie and specifically neuroscience. So our key common mission in AbbVie Neuroscience is preserving personhood. And that means making people able to maintain the feel of self, whether that's in neurodegenerative disorders, in psychiatry, in migraine, or in spinal cord injury. So I'm not going to go too much into that, but that's really makes us ensure that people living with these indications are the core of what we do every day. Okay, so I'm going to first give you an update today on our phase 2 spinal cord injury study. It's called ELASCI, which is elezanumab in acute spinal cord injury. Elezanumab is an inhibitor of an inhibitory process, and it is intervened in the first 24 hours after spinal cord injury. And it has been shown to have both neuroprotective effects, hence the need for it to be administered early, and neuroregenerative or enhancing plasticity, as Jim likes to say, in both rat and non-human primate models of spinal cord injury. And those data were just published recently. So elezanumab is administered via IV. Again, within 24 hours, that's what we saw in our preclinical data, had the best outcome as early as possible, but was translated to 24 hours in the human. And then every 4 weeks for 52 weeks in this initial study. We really wanted to interrogate the whole recovery process with this in the first of what we hope to be a few studies and the smallest of all those studies. So the ELASCI phase 2 study was initiated in 2020, so we went through the pandemic problems as well. It's really tough running any clinical trial in the pandemic, but particularly in an acute spinal cord injury trial where a lot of resources at hospitals were taken over to COVID. But we persevered, which I'm really pleased to say AbbVie and our leadership and our sites stayed on board. The study is targeting 60 acute traumatic cervical spinal cord injury patients across 6 countries, the US, Canada, Spain, Australia, Japan, and Korea. And I've been given the okay to share with you today that we completed enrollment last month. So we have finished the enrollment part of our study. Gets me a little choked up. After listening to everything that Rick, Jim, Steve said about the challenges in the acute spinal cord injury space, it still amazes me, even since 2020, that we were able to achieve this. So now we just need to retain those patients and wait for a year and get some data. So we are still completely unblinded, so I can't share anything about any treatment effects, and no one at AbbVie has any idea about study unblinding. The primary endpoint is the upper extremity motor score of the INSCI. We talked a lot about outcomes and the INSCI and challenges with INSCI. In acute spinal cord injury, it's really the best we've got. Honestly, what can you get at baseline within 24 hours that you can measure? Really hard to ask about quality of life and activities of daily living within the first 24 hours. So I'll talk a little bit about how we're supplementing the UAMS. So we're doing secondary endpoints that start later on after the injury, like the ADL, SCIM3, PsyQual, fluid and imaging biomarkers. And today I'm going to talk about electrophysiology. And does that really kind of go with this whole PFDD thing? Like, who cares what your muscles are doing with these little biosensors that I'll tell you about? The challenge not only with the UAMS during the acute phase is across a global site, multi-center study like this, you get such incredible rater variability on the INSCI. Even if you're using the same rater throughout the study, which rarely happens, we ask sites to do it, but you get turnover. And people who, if the participants want to come in on one day and somebody's not there, you're not going to tell the participant to come back another day when the rater's there. You're going to use another rater. So we are hoping to look for objective evidence of muscle activity outside of our clinical endpoints. So these are kind of some of our hypotheses, is that electrophysiology can be used as a biomarker of functional recovery and a potential objective evidence for muscle activity even prior to volitional movement. The detection of subclinical muscle activation where you can't actually visually see contraction. And then eventually we hope to correlate that with our clinical muscle assessments as well. We're doing this in two ways in the Alasky study, and this is what I'm going to talk about today is the wearable biosensors that we're doing at the sites in the United States. That is only because of the way that these biosensors are approved. They're FDA approved for use, but not outside of the United States. They're collecting both accelerometry and surface EMG via these biosensors. The biosensors are very scalable, and I'll pass them around here after this slide. You can distribute them to multiple sites. They take the data, they send it up to a cloud, and metadata, who owns the biosensors, can actually do the analysis from the cloud, which is really cool. And then in addition to that, we are also doing the classic electrophysiology at a single site in Spain, and they're doing motor evoked potential and surface EMG in the classical way. So we'll actually be able to look to see what kind of response are we getting via the biosensors and via classic electrophysiology. So let me talk a little bit about the biosensors. I'm going to pass a few out, and you can pass them around. Make sure they kind of come back to David or me. Yeah, David, go ahead. I'm going to show one first. So these little guys, they're like about the size of a bandage, and they have 4 electrodes on the back side, and they are applied to the individuals in the study on the upper extremities, 4 locations, right and left, and then the data are collected. Go ahead, you can pass this out. Thank you, my assistant, lovely assistant, David. On the bicep, tricep, rhomboid, and carpi. Okay, so these things do then there's a device and it looks like an iPhone, and you turn it on, it starts capturing data, and this is done during the manual muscle exam, and the participants are asked to do, try to contract your muscle 5 times. So, and they do that a couple of times during the actual assessment. So this is some examples of a clean signal from the bicep location. And on the top here, what you're seeing is the surface EMG sensor data during movement. And this is during a successful contraction. And then you actually see the raw data and the filtered data. And then we actually do some additional analysis so that you take out anything below 0. And you actually look at those data that are actually a squared version. And what you can see is on the top 3, surface EMG, you actually are seeing the 5 contractions. And then the bottom portion, the bottom thing here, is the accelerometry. And you can also see movement during that period as well. And I've got the names of our metadata analytics team. I do not do these analytics, so don't ask me about them. We will be publishing the data when they come out. Okay, so this is interesting. So this is what we do. If you look on the left-hand panel of data, you're looking at a successful contraction. And on the right, you're seeing a failed contraction. So like we saw in the example, the raw surface EMG, even within the raw data, you can see 5 distinct contractions, still a little noisy, and failed contraction, you just see noise. And this is all in volts or millivolts on the y-axis. Once you filter it, you could even see the cleaner contractions, single contractions in that filtered surface EMG, and again, nothing on the failed contraction. Then when we look at filtered and processed, you can actually see amplitude and, actually size of the contraction relative to each one of the successful attempts. And then again, you cannot see any 5 contractions on that failed contraction example. And on the accelerometry, you see some kind of distinct different peaks. Kind of hard to see within here because there's multiple different signals within the accelerometer, but that's measuring actually things are moving, right? And same thing again on failed contraction, it's just a lot of noise from the electrodes themselves. Okay, so this is an example of what we're doing is looking at the information I just showed you over a period of time. This is an example data only. So if you look on the left-hand side, I'm showing you accelerometer data on the top from each one of those biosensor locations. And on the top, it's accelerometry. And let's take example of the biceps. You don't see anything in accelerometry, so that means you're not seeing any kind of movement at all from the sensors themselves until week 36 and week 32. So green is a positive signal. Same thing with the rhombus right and rhombus left. So if you look down to the surface EMG, and this is what we're seeing, but I can't show real data yet. My IP attorneys guided me not to do that. But this is an example of what we're seeing. We're seeing some muscles that actually are showing surface EMG prior to detection of accelerometry. So we're seeing earlier actual indications of muscle activity, and then later on we're seeing that actual increase into the ability to move via the accelerometer data. And then what we want to do next is say, well, how does that correspond with what we're seeing in the clinical assessments in the UAMS? Are we seeing UAMS at the same time we're seeing some muscle movement and contraction, or are we seeing it earlier? So that's our primary endpoint, the UAMS, so I can't talk about that quite yet. So finally, in summary, what we're doing with this is we can observe and quantify changes, so this is more objective, gets us a little bit away from the rater variability as well, muscle function and accelerometry and surface EMG. We can actually capture the amplitude as well, so we can measure not only can they do it, but with what type of strength are they moving. And we think that this might be more sensitive than the surface EMG and accelerometer are more sensitive than that upper extremity motor exam, not just due to rater variability, but because of the exam itself. So there's a couple considerations we thought of in reviewing these data. So the UAMS is a different exam than the manual muscle test. So you might be some subtle differences between those two exams based on how the assessment is done. So if there are differences, it might not just be due to the biosensor itself. Surface EMG and accelerometry, they are objective. And some other things that we found is it's really hard to disconnect where the surface EMG signal is coming from and separate that from the accelerometry. Right? If you can think about it, you're moving your arm and you're also contracting your muscle. And maybe that's okay. Maybe those are just the data that we have because we've got muscle movement and EMG. If we would have thought about it, or if I would have thought about it before, actually doing some isometric exercise may have been better because we're not actually trying to record movement at the same time as trying to record the surface EMG. So our next steps are to compare surface EMG and accelerometry to our primary endpoint, see if we actually have a biomarker of do we see surface EMG, then we actually see earlier, then we see UEMS, and it could be predictive and objective. And then we want to compare the surface EMG, to the motor evoked potential data that we're doing in our electrophysiology sub-study as well. So are these biosensors picking up sensitive assessments of surface EMG? And then I added this today because I listened to the panel intently and was very moved, as you've heard many people. And I really want to find out from the participants in the study, like, what was it like having these biosensors placed all over your body? Was it intrusive? How did you feel? Did the sites adequately explain to you why we were doing that? We train sites to do that, but there's turnover, and we train them once. We need to keep training and saying, this is why we're doing this. This is to find out what's happening with your body. You're a co-researcher. So I hope that helps a little bit in sharing with what we're doing right now, and I hope maybe this time next year, a little later than this time next year, we'll actually have data from the study, and I'm super excited to share with that as well. Thank you.